Today I'm going to talk about the range of thallus organization in algae. Algae show a considerable variation in the organization of thallus and is broadly classified into 5 forms. That is unicellular forms, colonial forms, filamentous forms, siphonous forms, parenchymatous forms. First of all, unicellular forms. The unicellular forms divide and result in the formation of multicellular forms. In unicellular form, the division takes place that result in the formation of multicellular forms. The unicellular forms of algae are seen in all the groups of algae except the Charophyceae and Phaeophyceae. These forms are found in all groups of algae. Except Charophyceae and Phaeophyceae. These unicellular forms are referred to as acellular forms. They are referred to as acellular forms. They function as a complete living unit without any cellular differentiation. Acellular forms means the complete living unit without any cellular differentiation. These unicellular forms are further classified into 4 subgroups. They are further classified into 4 subgroups, such as rhizopodial unicells, flagellated unicells, spiral filamentous unicells, and non-motile unicells. These are all unicellular. First of all, rhizopodial unicells. The rhizopodial unicells, these forms lack the rigid cell wall. They possess cytoplasmic projections which helps them in amoeboid movement. These cytoplasmic projections helps them in amoeboid movement. Means they move by these cytoplasmic projections. Then in the absence of these rigid cell wall, they form periplastic. In absence of cell wall, they form periplastic. The example of rhizopodial unicells are rhizochrysas, rhizochloris, etc. Then second is flagellated unicells. Flagellated simply means they possess flagella. The vegetative cells possess flagella, and these forms are, they possess flagella, and these forms are found in all groups except Cyanophyceae. Except Cyanophyceae. Then we have spiral filamentous unicells. The spiral filamentous unicells form spiral or coiled structure. They are spirally coiled structure. Coiled unicells. Example, spirulina. In case of flagellated unicells, example is Chlamydomonas. After spiral filamentous unicells, we have non-motile unicells. Simply non-motile unicells. These are simply non-motile, means they do not possess flagella. They don't possess flagella, eye spot, etc., that are meant for locomotion. The simplest non-motile forms are found in Cyanophyceae. Simplest forms are present in Cyanophyceae. Where they do not possess the plastids and nucleus. But in case of Chlorophyceae, the nucleus and plastids are present. Then we have colonial forms. Second, colonial forms. What are colonial forms? The colonial forms are formed by the aggregation of products of cell division within a mucilaginous mass. Therefore, all the members of the colony may have the similar structure. All members of colony have similar structure. Similar structure. These associations are usually loose, and as such, some colonies break into smaller pieces. But in some colonies, like we have the Volvox colony, the all members of the colony are connected with each other by cytoplasmic strands. In Volvox, all the members of colony are connected to each other by cytoplasmic strands. They are connected to each other by cytoplasmic strands, and hence they cannot break into segments. Example, Volvox. On the basis of morphology, the colonial organization may be divided into 4 types. On the basis of morphology. On the basis of morphology, the colonial forms are further classified into 4 subclasses. or subgroups. That is Cenobium, Palmilloid, Dendroid, and Rhizopodial. First, Cenobium. In the Cenobium, the colony possesses a definite shape, size, and arrangement of cells. Definite shape, size, and arrangement of cells. Arrangement of cells. The number of colony in a cenobium is determined at the juvenile stage, and subsequently there is only increase in size. These cenobial colonies may be motile and they may be non-motile. Motile or they may be non-motile. Means either they possess the flagella or they don't possess the flagella. Motile forms, in motile forms, the example is volvox, and in non-motile forms the example is hydrodictio. It's non-motile form. Then second we have palmilloid. These colonial algae are filamentous and are externally covered by mucilage sheath. And are covered by mucilage sheath. These colonies do not possess a definite shape and size as in case of cenobium. So they don't possess definite shape, size, and number of cells. The cells of the colony are independent. The cells of the colony in case of palmilloid are independent. Example, Chlamydomonas. Then third we have dendroid forms. Dendroid forms, these are also the colonial forms. These colonial algae resemble the palmilloid algae, but the mucilage thread is present only at the base of each cell. In case of dendroid, the mucilage thread is present only at the base of each cell. And the threads of different cells are united to form a branched structure which give a tree-like appearance. The threads of different cells are united and they give a tree-like appearance. They form a branched structure which give a tree-like appearance. Example, Chrysodendron. Then fourth one is Rhizopodial. This is the fourth one. These rhizopodials, these are also the colonial forms, and the cells are united by means of rhizopodia. Means in this colony, cells are united, attached to each other by means of rhizopodia. Then we have filamentous forms. Filamentous forms. Filamentous forms are developed by the repeated transverse division. Here, repeated transverse division of cells takes place. Repeated transverse division. Division of cells takes place, which result in the formation of these filamentous forms. The daughter cells do not separate but remain attached to the parent cell or to one another in a definite sequence to form the filaments. Daughter cells do not separate and give rise to filamentous forms. The filaments may be branched or they may be unbranched. Branched or unbranched. In branched filaments, these branched filaments are formed by the repeated division of lateral outgrowth of the cell. Lateral outgrowth of cells undergo repeated transverse division. And hence the branched filaments are formed. But in case of unbranched filaments, the simple unbranched filaments are formed and they may be free floating or attached to the substratum. These simple unbranched filaments are found in few groups of algae. Unbranched, they are present in few groups of algae and may be free floating or attached to substratum. And finally, the fourth one, we have siphonous forms. Siphonaceous forms. Siphonous forms. In case of siphonous forms, a repeated nuclear division takes place. Here, repeated nuclear division takes place. Repeated nuclear division takes place without cross-wall formation. Which give rise to the siphonous forms. The repeated nuclear division takes place without the cross-wall formation, which give rise to these siphonous forms. And here the thallus is made up of aseptate tubular filaments. It resembles with the cenostic condition seen in fungi, in which the nuclear division are not accompanied by the wall formation, and therefore the content of the cell intermix so as to form multinucleate mass of cytoplasm. Multinucleate mass of cytoplasm is formed. Then fifth we have parenchymatous form. In this type, flat folios or tubular thalli are formed. Either flat folios or tubular thalli are formed. These tubular thalli or the flat folios are formed by the division of cells in two or more planes. In two or more planes. The daughter cells do not separate and give rise to filamentous thalli of various shapes. Daughter cells do not separate and give rise to parenchymatous forms of various shapes. Such as flat, tubular, complex, etc. Example, we have fucus, sargassum, laminaria, alva, etc. In case of siphonos form, the example is Vacheria, where the nucleus undergo numerous nuclear division result in the formation of a multinucleate mass. Example is Vacheria.